Good morning, everybody. I am just going to pull up the PowerPoint and then we'll get started. There is actually not a whole lot of content that we really need to go over, because if you've looked at all at the two outlines that I posted, first of all, for the reproductive and development section, and then for heredity and genetics, there is already a lot of information filled out for you on there. So because you have a lot of it already in there, there's not a whole lot that we really need to go over, but I am going to go over what isn't in the outline already or on the web pages that I've given you in D2L. So here is the development and genetics PowerPoint. But I am going to start first with the chart that is in the reproductive system and development outline. It starts on the bottom of page 12. If you have the outline in front of you, or if you can pull it up on your computer, this is the chart that is part of the female reproductive cycle. It outlines the days of the reproductive cycle, and it breaks down the events that happen during the ovarian cycle, the ovarian portion of the reproductive cycle, and then the uterine cycle. So what's happening within the uterus during those times. So there's a few things in there that aren't completed in your outline, so I'll help you fill that in. But pretty much everything else, if you look through the reproductive section, it is either filled in for you, or I have the information within the web page, the reproductive system web page that I made in D2L. You have everything in there. So this is the first thing that is not provided for you. So like I said, it's at the bottom of page 12, and I will start with just a quick overview of the reproductive cycle, and then we'll fill in that chart. So this occurs over a period of roughly 28 days. It does vary slightly from person to person. And then it involves events that are happening within the ovaries during that time, and then simultaneously events that are happening within the uterus. So we'll look at the ovarian cycle first. During the follicular phase, which is basically the first half of the reproductive cycle, it's like day 1 all the way up to day 14 is the follicular phase of the ovarian cycle. So what's happening at that point in the ovaries is that follicle stimulating hormone is secreted, and it causes tertiary follicles. So when we say follicles, hopefully you've read a little bit at this point, so you know a follicle is the egg, the female egg, the ovum surrounded by cells. So when that hormone is released, it causes tertiary follicles to grow. And then what's already in the chart is that one of those follicles is going to become dominant, and the primary oocyte, which is the egg, within that follicle is going to complete meiosis 1. So that is part of cell division that's happening. And that forms a secondary oocyte and a polar body. So it's dividing into 2 separate cells, forming the secondary oocyte and the polar body, which is just discarded material. Then a second hormone gets secreted, luteinizing hormone, and its release stimulates ovulation. So once that hormone is released, it triggers ovulation. So that's what actually happens next in the reproductive cycle. During ovulation, so this is the next row on your chart, the tertiary follicle ruptures, and it releases the secondary oocyte. So an interesting thing can happen at this point. Fraternal twins would develop if 2 oocytes are ovulated. Normally it's just 1, but if there are 2 that get ovulated, and then if both are eventually fertilized by 2 different sperm cells, then that results in fraternal twins, not identical twins. So in this case, there are 2 separate oocytes and 2 separate sperm cells. Okay, so that's what happens during ovulation. The oocyte gets released. And then the last phase is basically the second half of the reproductive cycle. It's called the luteal phase. So at this point, the tertiary follicle, what's left of it, because now it's released to the oocyte, it collapses, and now it's called the corpus luteum. And that secretes estrogen and progesterone. Those are 2 hormones. So the corpus luteum secretes estrogen and progesterone. And at this point, there are 2 different things that'll happen depending on whether or not fertilization of that secondary oocyte that was ovulated, whether fertilization has occurred. So if there is no fertilization, then after about 10 days, the corpus luteum degenerates. So it kind of, it gets smaller, and it becomes the corpus albicans, and it stops secreting hormones. If, on the other hand, fertilization does occur, then the corpus luteum is not going to do that. The corpus luteum is going to persist. It's going to remain there, and it will continue to secrete hormones until the placenta is formed. All right, ultimately, the placenta will take over that function of secreting those 2 hormones. But until that time, the corpus luteum will continue to secrete estrogen and progesterone. Okay, so we filled out that chart, and now we can go down to just onto the next page, where there is further information about human development. So this is kind of picking up right at fertilization and explaining, first of all, what happens during fertilization, and then all of the development that happens immediately following fertilization. So I'm going to just fill in the sections that aren't already filled in for you. So just scrolling right down to the embryonic development section at the top of your page 14. Basically, embryonic development, it refers to the first 8 weeks or 2 months of development, and then fetal development is the rest of development. And embryonic development begins with fertilization. That's when sperm genetic material combines with the oocytes genetic material. It usually occurs maybe a full day or within a day after ovulation. And you have the steps of fertilization listed for you. So now we do have that in the PowerPoint. You can see on these pictures here, they're showing you the steps that occur during fertilization. So first of all is capacitation, and that refers to the functional changes in the sperm that make them more mobile, so able to move, and allow them to penetrate the oocyte's plasma membrane. So after that happens, then you have the acrosomal reaction and penetration. That involves the release of acrosomal enzymes that are used to penetrate the layers surrounding the oocyte. Also, during this time, you want to prevent polyspermy, which refers to fertilization of the oocyte by more than 1 sperm. If that happens, then you will not have a viable embryo, and it won't continue to develop. So you want to prevent other sperm from being able to get in and fertilizing the same oocyte. So there's two things that help with that. And I believe there's, oops, no, I want to go back. Yeah. This picture kind of helps to explain one of them. So there's a fast block, which occurs very quickly, and then a slow block, which takes a little bit longer, but it's more permanent. The fast block, and these are explained already in your outline, but that's when the oocyte's receptors that bind to the sperm are actually shed. So that prevents other sperm from being able to bind to the oocyte. And then the slow block involves basically the hardening of the layer, which you can see here, the layer that surrounds the oocyte. So after that occurs, then you have completion of meiosis 2. That will only occur once the sperm fully penetrates the oocyte and the secondary oocyte becomes an ovum. And then actual fertilization occurs once the chromosomes from the sperm and the ovum combine. So that restores the normal chromosome number, which is 46 chromosomes. The cell that results, the single cell that results from that combining, is called a zygote. And that's what you start with during week 1 of embryonic development. As soon as you have the zygote, then there is a lot of cell division that happens right away. That's called mitosis. So there's very rapid cell division. And the term blastomeres refers to all of the smaller cells that are produced by that cell division. If you're looking at this picture or these pictures on the PowerPoint, all of these individual cells are blastomeres. Same thing here. All these cells are blastomeres. The term morula refers to the solid ball of blastomeres. Okay, right here is a morula, a solid ball of cells. So as you're going through cell division, the morula slowly moves through the uterine tube to get down into the uterus, because that's where ultimately it's going to implant and then develop. At that point, once it reaches the uterus, fluid from the endometrial layer of the uterus goes into the morula, kind of pushes the blastomeres off to the side, and that forms a blastocyst, which you can see on this third picture. The blastocyst is the term for the morula after it develops this fluid-filled cavity in the middle. And now there are two distinct layers that are described in your outline. There's the trophoblast, which is this outer layer of cells, and the embroblast, which is the inner cell mass here. Okay, and then after that, implantation occurs, which is when the blastocyst attaches to the endometrium. So that will happen after about a week, 7 days after fertilization. And you have a description in your outline of what occurs during implantation, tells you where it occurs. You can see that here. It's typically in the body of the uterus. So that is at the end of week 1. By the end of week 1, you've had implantation, and now we can start week 2. So during week 2, this is when a lot of membranes develop that are associated with the embryo. The first one is called the amnion. Let me go to the next slide. Here's a picture of implantation. So during the second week of development, we have formation of the amnion, which is a thin protective membrane that surrounds the entire embryo as it grows. And within the amnion, you have an amniotic cavity that's filled with amniotic fluid. So that surrounds the embryo. Also, there's development of the yolk sac that develops from the blastocyst cavity, which is here. And this is actually where blood cells are produced for the fetus. After birth, you no longer have a yolk sac, and then blood cells are produced in red bone marrow, if you remember. Okay, another one is the allantois. This is a vascular pouch in the wall of the yolk sac. Ultimately, this forms part of the umbilical cord. And then the last membrane that develops at this time is the chorion, which develops from the trophoblast, which is that outer layer of cells. The chorion forms the embryonic portion of the placenta. So this is one of the layers of the placenta. Okay, another big thing that's happening during this time is formation of the placenta. That is a temporary organ that allows the exchange of substances between the mother and the developing embryo. The embryonic portion of the placenta, remember we just said this, is the chorion, and the maternal portion is formed from the endometrium. Remember, that's the inner lining of the uterus. So here's a good picture showing a more developed embryo right here. The amniotic cavity, the amnion containing the amniotic cavity that surrounds the embryo. Here's the yolk sac. Here is the allantois, that's going to become the umbilical cord, remember? And then all the way outside is the placenta. Okay. Going down in your outline, you can see that during week 3, you have the process of gastrulation occurs, which is when the embryo develops 3 primary layers, which are ultimately going to become specific things in the body, specific tissues. So gastrulation produces what's called a gastrula. And during that time, you develop these 3 layers, which are listed on the left side of the chart. You have ectoderm, the outer layer, mesoderm, which is the middle layer, and endoderm, which is the inner layer. The ectoderm is ultimately going to become the epidermis and the nervous system. The mesoderm, which is in the middle, is going to form all of the connective tissues within the body. So that involves things like cartilage, bone, adipose tissue, blood even. The mesoderm also will form muscle tissue, blood and lymphatic vessels, and the lymphoid organs, things like tonsils, spleen. Okay, and then finally the endoderm, which is the inner layer, will form the lining of all of your ducts and your tracts. So it'll form the lining of the digestive tract, respiratory tract, urinary tract. Okay, also a really important thing that happens during week 3 is formation of the neural tube. This is when folds form, and I think I may have a picture of this, yep. Folds form in the ectoderm. So if you look at the top of this middle picture here, you can see that happening. So these folds develop in the ectoderm, and then eventually they fuse together at the top, which forms the neural tube. The neural tube cells develop into the brain and the spinal cord. So this is a very important step in development that's happening. If the neural tube doesn't develop properly, that causes a neural tube defect. And some of these include spina bifida and anencephaly, which you may have heard of. Spina bifida typically is not fatal, but anencephaly is. Okay, so that's all during week 3 of development. If we go to weeks, just the second half, so weeks 4 to 8, which takes you through the end of embryonic development, this is when you have a lot of organogenesis, which is the differentiation of those 3 layers that we just talked about to form organs. So by the end of embryonic development, once the time you reach week 8, all organ systems are developed. So you have all of your major organs developed. They may not be functioning, but they have formed. At this point, the embryo is still very, very small. It's slightly over an inch and it weighs like 2 grams. It's very small. And after that point is fetal development. That's the last 7 months of development. During this time, it's a period of really rapid growth, but there's not a lot of new structures that form because pretty much everything was developed during embryonic development. So you just have a lot of growth of the structures that were already formed during embryonic development. Some things that happen here, let's just talk about fetal circulation, so blood vessels that have developed. The fetus does contain a few modifications that you only have during prenatal development. After birth, these things disappear. So during the prenatal time, before birth, there are umbilical arteries and an umbilical vein. The umbilical arteries are paired vessels that carry blood from the fetus to the placenta. And the umbilical vein is a single vessel that carries oxygenated blood from the placenta into the fetus. So that is how the fetus is getting oxygen. And then also there are 3, they're called vascular shunts. So they're just carrying blood from one place to another. But these also don't exist after birth, they go away. There is one called the ductus venosus. This carries blood from the umbilical vein. It goes past the liver and into the inferior vena cava. Then there is another one called the foramen ovale. This diverts blood from the right atrium directly into the left atrium. So it's actually a small hole in between the 2 atria. And then finally, the ductus arteriosus, that diverts blood from the pulmonary trunk to the aorta. Okay. So after birth, these umbilical blood vessels and shunts are not necessary. A lot of them just kind of collapse and they turn into ligaments, which are just like cords of connective tissue. And then the foramen ovale closes up. That's the hole in between the 2 atria, and that closes. All right, so that takes us to the end of the reproductive and development outline. And now we can go into heredity. So if you go into that outline, starting at the beginning, there's just a lot of terminology there. I think, yeah, this PowerPoint just continues right on. So you have definitions for a lot of the terms already. The 2 that you don't have defined are referring to alleles. So you have to understand what an allele is. It's a copy of a gene. So it's like 1/2 of a pair of genes that you have. And if we refer to them as being homozygous, that means that there are 2 identical alleles for the same trait or for the same gene. So in that case, if you have 2 of exactly the same alleles for a particular trait, then you're always going to see that trait. So for example, if you have 2 alleles for, let's say, blue eyes, then the individual is definitely going to have blue eyes. An alternative option is if the alleles are heterozygous, that means that there are 2 different alleles for the same gene or trait. So an example would be if you have 1 brown allele and 1 blue allele. How do you know what color the person's eyes are going to be? Well, usually one allele is dominant over the other one. So one is dominant, the other one is called recessive. And the dominant one is the one that is expressed. That's the one that you see physically. The recessive allele is not seen at all. And you would be told, if that is the case, you would have to be told which one is dominant and which one is recessive. There's no way of just knowing that. So in the case of eye color, if you have one blue allele and one brown allele, brown is dominant, blue is recessive. So that means what you're physically going to see in that individual is brown eyes, because brown is the dominant allele. Okay, so going down a little bit further, I am going to explain a Punnett square and then just the different types of inheritance. And then that's it. That takes us through that outline. So this is going to be kind of short today because a lot of, like I said, a lot of the stuff is filled in for you guys already. So for the Punnett square, it's used to determine the possible gene combinations. Hold on, I'm going to skip through this real quick. The gene combinations for potential offspring if you have two individuals that are having offspring together. Let me go back. All right. So that's based on just the normal dominant recessive inheritance, which is when one allele is dominant, the other one is recessive, and it's the dominant one that you see. So to do a Punnett square, you will be told what the two parents are, what their genetic makeup is. So that's called their genotype. What alleles do they have for a particular gene? And then you'll use that to determine the possible allele combinations for their offspring. And from that, you can figure out what physical traits the offspring might have. So in this example, you have a father that is heterozygous for a trait. And you have a mother that is also heterozygous. So you have to remember what heterozygous means. It means that there are two different alleles. So that means one allele is dominant, the other allele is recessive. And the way that you represent, if you're like writing this out in a Punnett square, the way that you represent the dominant allele is using a capital letter. So they've used a capital P here for the dominant allele and a lowercase p, which represents the recessive allele. Okay. For the mother, it's exactly the same. She is heterozygous, so she has one dominant allele and one recessive allele. So to set up the Punnett square, you make a square, like you can see here, with 4 boxes, 2 on the top, 2 on the bottom. You're going to separate the 2 alleles from the one parent. It doesn't matter which one you put on the top and which one you put along the side. They have put the father's 2 alleles along the side. So they put the capital P and the lowercase p here. And then for the mother's 2 alleles, they put them along the top. So capital P is above this first column of boxes. Lowercase p is above the second column. And then what you do to figure out the possible allele combinations is you just bring the letters across and then down. So you will bring the capital P across into this box and this box. So there's one capital P, one capital letter P here. You bring the lowercase p across into these 2 boxes that it's next to. So there's a lowercase and a lowercase. Then for the 2 letters that are on the top, you bring them down into the 2 boxes that they're right above. So capital P comes down into this box and down into the second one. The lowercase p comes down into the first box, down into the second one. And now you can see, this is filled out already, you can see the possible allele combinations for the offspring. So 4 out of the 4 possible offspring, 1 would be capital P, capital P. So 1 would have both dominant alleles, 2 dominant alleles. That's 25% of the possible offspring would have 2 dominant alleles. Another one would have 2 recessive alleles. So 25% of the offspring, there's a 25% chance basically that the offspring would have both recessive alleles. And then 2 of the offspring out of 4, 2 out of 4 would be heterozygous with 1 dominant allele and 1 recessive allele. And this one is the same. So there's a 50% chance that the offspring would be heterozygous. Okay, we can do a different one that I'll write it in just to give you some practice. Okay. So here is, it's not going to look perfect, but here's the Punnett square. And we're going to say that the father is, well, let's say this is to figure out eye color, first of all. And we'll say that the dominant allele, so that has a capital letter, codes for brown eyes. And then we're going to say that the recessive allele is blue eyes. And what if you have a father who has blue eyes? So he must be homozygous recessive. That means he has 2 recessive alleles, 2 little lowercase b's for blue eyes. And we'll say that the mother is homozygous dominant. That means she has 2 brown alleles, 2 dominant alleles. That gives her brown eyes. What are the possible offspring? So we'll do 1 across the top. We'll put the mother on top and put both of her alleles above each of these columns. We'll put the father on the side, both of his alleles next to each row. And then we just bring those letters down. So down here, coming down into this first column, we'll have a capital, whoops, let me do this. We'll have a capital letter B coming down here and then down here. Same thing above the second column. We're going to bring down the capital letter B into both of these boxes. And then we go to the father's side and we bring across the lowercase b into both of the boxes that it's next to. And we do the same thing for the bottom row. Bring across that lowercase b, that recessive allele. And now these are the possible offspring. You can see that they're all the same. There is 100% chance that the offspring will be heterozygous. That means that all the offspring, the only chance if they have offspring, they will have to be heterozygous. That means they'll have 1 dominant allele and 1 recessive allele. So 1 allele for brown eyes, 1 allele for blue eyes. What is their eye color? Remember, the dominant allele is the one that's expressed. The recessive allele you don't see. So the eye color for heterozygous will be brown because the brown allele is dominant. So there's 100% chance that their offspring will have brown eyes. If you're asked, what are the chances that their offspring will have blue eyes? It's a 0% chance. They just can't.
Why is blue recessive?
Because it just is. Like I said, you'll be told which one's dominant and which was recessive. That's just how the genes work. So one is dominant over the other.
Okay.
It's not something you have to figure out. It's just the way that the way the chromosomes interact.
So you know this, or you just was saying that for the sake of this problem?
No, that's true. And you would be told. You'll always be told which is dominant, which is recessive, because there's no way for you to figure it out. It's just a known thing.
Okay, let me ask you this. When you put the two bees, the dominant bees in the blocks first, is that because they're dominant, so you listed those first rather than bringing the little bee over for dad first?
Yeah, typically the way that it's written out is that you always put the dominant allele first.
Okay.
If you did it the other way, I wouldn't mark it wrong, but that is just the typical way that it's written. You write the dominant allele first.
So if we had written it the other way, then that would have made it like the recessive one would be 100%.
No, writing it the other way. So whether you write it, I'll do it. Whether you write it with the dominant first and the recessive.
No, the opposite.
Or, well, I'm just giving you an example.
Oh.
So if you write it this way, or if you just switch it
and
you write it that way, it's still the same, right? You have a dominant allele and you have a recessive allele.
Right. Okay.
The dominant one is always the one that you see.
Okay.
So no matter how you write it, you could write it either way, but you still have the same two alleles and they still interact the same way. The dominant allele is the one that's going to like take over. It dominates. So that's the one that you see.
Okay.
So it doesn't matter how you write it.
Okay.
You still are going to just see the dominant allele, which in this case is for brown eyes. So either way, that person would have brown eyes.
Okay.
Okay. Thanks. Good questions. Yeah. Good questions. All right. I can give you guys some practice. Like I do have a practice handout for Punnett squares because there will be 2 that you'll do on the test. You don't actually have to write out everything, but you will have to like type in your answer for what you get. So you will actually have to do it like on paper in front of you, unless there's some way you can do it in your head, which would be difficult. You don't have to show the Punnett square, but you will type in your answer. So those questions will require that you do a Punnett square. And so I can post on D2L some practice Punnett squares just to give you some extra practice on those.
Gotcha. I have another question.
Yeah.
As far as the extra credit questions, how are we presenting those to you? Because I didn't see those on the last test, but I know there was some on the
I took that up. So I just had an incorrect test review posted. If you look, I removed it. So there's not. Yeah.
About the one that's upcoming. Are you speaking on that one or the old one?
Both.
Oh, okay.
I corrected it. So they're both like the one I don't have posted yet for this test. For the last one, I did have an incorrect test review posted and I corrected it. And I did send out an announcement letting you know that it changed so that the correction was made. Yeah, sorry about that. So you will have a test review. I'm going to put it up really soon and it'll, there's not extra credit there.
Gotcha.
Okay. All right. So if no more questions about the Punnett square for now, I'll just keep going through and really just its definitions for the other types of inheritance. So by far, the most common type of inheritance is the dominant recessive type that we just talked about, where there's always one allele that dominates over the other one. That's the dominant allele. The other one is the recessive allele. And it's the dominant one only that you see.
Mhm.
But there are some other types. There is another type of inheritance that is called incomplete dominance. With this type, neither allele is dominant over the other one. So if you have one of each, if you have a heterozygous genotype, which is where you've got one of each, you have one dominant and one recessive, what you'll actually see as a result is a combination of those two alleles. So I'll give you a good example here. Oops, let me get rid of, hold on, all that writing. Okay. So here's a good example. Like in flowers, you can have red. So there's an allele for red flowers. And there is an allele, so capital R for the red, lowercase r for the white. But actually, in this case, if it's incomplete dominance, neither one is dominant over the other. So if you have two of these, that would give you red flowers, because you have two alleles coding for red flowers. So you're definitely going to have red flowers. If you have this, then that lowercase r gives you white flowers. So if you have two alleles for white flowers, you're definitely going to have white flowers. But in this case, if you have one of each, what we just learned earlier was that one is dominant over the other. So the dominant one is the one you're going to see. However, this is different because in this case, neither one is dominant. So if you have one of each, what you're going to see is a combination of the two. So if you put one red and one white together and you mix them together, the result is pink, like an intermediate between red and white. So that's not very common, but that does exist.
Wow.
So that's a different type of dominance. Incomplete dominance is where neither one is dominant over the other. So if you have one of each allele, what you see is a combination of the two. Okay.
Interesting you say that, because I have a friend, her daughter has two different eye colors, but they call the other one a birthmark. So one is like hazel, the other one is dark brown.
Yeah. So that is actually, that's not this.
It's not.
Yeah. Eye color is dominant recessive.
Okay.
That is determined by dominant recessive. So that likely is because eye color is determined by the amount of melanin that you have in your eyes. Melanin is a brown pigment. It's what gives skin its color too. So people that have darker skin have more melanin. People that have really light skin have less melanin. There's all kinds of variations. So that can happen where you do, like it is like a birthmark, where you have a patch of skin that contains more melanin. That can happen in eyes as well. So that is truly what happened with her eye, probably. Like you have just an area where there's more melanin, so it's darker.
Oh, wow. Okay.
Yeah. So like I said, incomplete dominance is really rare, but you do have it with a couple of characteristics with humans. One of them is sickle cell anemia. So you can you have the allele that causes sickle cell anemia. You can have the allele that that gives you just normal red blood cells or normal hemoglobin. And then, but then if you get one of each allele, one that's for normal hemoglobin and one that causes sickle cell anemia, if you have both of those alleles together, then you have a combination. So you have some normal hemoglobin and some hemoglobin that's abnormal. So it's interesting. That's really one of the only examples where you ever see this type of dominance in humans.
Wow.
Okay, then there's just a couple of others. There's the next one. Let me go to the next slide. Whoops, hold on. Let me get rid of this. All right, the next one is multiple allele inheritance. This is when genes have more than 2 alleles. So a good example is with our blood type. With the A, B, O blood types, there are 3 different alleles, one for A, one for B, and one for O. And your blood type results from whatever combination, whatever 2 alleles you happen to get. So when there are multiple alleles involved, there's more possibilities, right? So instead of just 2 alleles and 2 options for what you see, what physical characteristic you see as a result, if you have multiple alleles involved, then there's more possibilities. So there's 4 possible phenotypes, 4 possible blood types that you can have, because there are 3 different alleles rather than just 2. Okay, so blood type can be, and we just talked about this, your blood type could be type A, type B, AB, or O. Okay, another one is polygene. Here we go, polygene inheritance. Actually, I'm gonna not go to that slide just yet. With polygene inheritance, this involves traits that are controlled by more than 1 gene. And then within each gene, there are at least 2 alleles. So there are many genes and therefore many, many different alleles that are involved in determining these traits. So these are the traits, if you think about it, these are the ones where you have like many, many different possibilities. So if we're talking about color, hair color, eye color, skin color, if you think about it, there's many possibilities, not just 2, not just 4, like with blood type, there's like tens upon tens of different options here. So it's those types of traits that are definitely controlled by polygene inheritance. There's multiple genes involved. Okay, which gives you that wide variety that you see. Height is another thing that's controlled by polygene inheritance. All right, and then one last type of inheritance is sex-linked inheritance. This is when traits are determined by genes on the sex chromosomes. So there's 23 pairs of chromosomes that we have. One pair are the sex chromosomes. So if you have, and I can go back, because I think I have a slide. Yeah. So your 23rd pair of chromosomes is what determines your sex, male or female. If you have 2 X chromosomes, you are female, genetically female. And if you have an X and a Y chromosome, genetically you are a male. Okay, the X chromosome is larger than the Y chromosome, as you can see on this picture here of all the chromosomes. The X is larger. So the X chromosome contains more than 1,000 different genes on it. There's just room for more. The Y chromosome is a lot smaller. It only contains about 200 genes. So therefore it lacks many of the genes that are present on the X chromosome. So if you are male and you have 1 X and 1 Y chromosome, So on your X chromosome, you're going to have a lot more genes than you have on the Y chromosome. So there's a chance that you only have one allele within a gene because you simply don't have it on the Y chromosome. Like it's not big enough. So you just don't have the gene on that. So basically any gene that is found on an X or a Y chromosome is considered whatever like trait it codes for is considered a sex-linked trait because it's found on one of the sex chromosomes. And then genes, because there are a lot, genes that are found only on the X chromosome because the Y chromosome just doesn't have them because it's too small. These are called X-linked genes. And there are genes that actually cause some bad things that are X-linked. An example, and the first one's not really a big deal, but the first one is color blindness. The most common type of color blindness is red-green color blindness, where you can't distinguish between those two colors. That is actually an X-linked trait. So there's a gene for that on the X chromosome, but there's not a corresponding gene for it on the Y chromosome. So that is one example. And then another one is the gene for hemophilia, the bleeding disorder. That is also only found on the X chromosome. So any gene that's found on either of the X chromosomes is controlled through sex-linked inheritance. All right, and that takes us to the end of that outline. So it was pretty short today because I gave you, like I said, I gave you a lot of information already in the outline. A lot of stuff was already filled in. A lot of the stuff for this test is just knowing the reproductive structures. And so definitely all of the definitions and descriptions for those structures are already in the outline. So go through that and use the pictures in the PowerPoint to help you identify where those structures are located. By doing that, that's also going to help you prepare for the final lab practical, because that's a lot of the information that is on the final practical. So definitely do that. And then you have just the development section, which we went through a lot of that today. And then the inheritance genetics section is pretty short. So not a whole lot of questions on that. A lot of it is just definitions. So knowing the definitions of the terms that are in the beginning of that outline, and then knowing the different types of inheritance, and being able to do a Punnett square, which there's 2 questions that are based on you doing a Punnett square and then answering the questions based on that.
Okay, so this test is on the 5th.
The test is, yeah, the 5th. That's next Thursday, right? Your test is next Thursday. Let me check the date. Yep, it is the 5th. So group B has lab on Tuesday of next week, just mainly to go over some of the models and the charts for the reproductive system and to review for the final lab practical.
Okay. Did you put pictures up for those, like the different models that we're going to go through? I didn't.
Yep. If you go to the final lab practical folder in D2L, they're in there.